Yes. Thank you for the introduction, Laszlo. Hiromasa, thank you so much for inviting me here. I want to talk today about, receptors as the link between structure and function. And most people, when they think about connectivity in the brain, what comes to mind is what you see here on the screen. It's fiber tracts, okay? But what they forget is that at the end of the fiber tract, we have an axon because neurons are not physically connected with each other. And at the end of this axon, at the synapse, we've got extracellular space, and there is the need of a second mechanism to transfer information in the brain. The first mechanism would be the electric one. That's the way that the signal is transported along the axon, okay? But when you reach the synapse, you need what is called chemical conduction. And that means that the presynaptic neuron, the one that has, my mouse. Okay, so the presynaptic neuron, the one that has information that has to be passed on, releases neurotransmitters into the... Oh, okay. Right, I hope this is better. Thank you. Yes, so neurotransmitters, these little green balls in this slide are released into the extracellular space, and they bind onto receptors on the target neuron. The receptors are indicated here by the pink cylinders. Now, it's important for you to know that in vivo, every single neuron can be targeted by very many other neurons, okay? And that each one of these different neurons can release a different kind of neurotransmitter. So it is very important that a given neuron on its membrane expresses receptors for very many different kinds of neurotransmitters. And that is the case that we have in vivo. Now, these receptors are simply either proteins or protein complexes, and we've got different methods that can be used to visualize them. Here on the left and on the right of the slide, we've got the two spatial ranges that we can cover with the methods. On the left, there's positron emission tomography, which has the lowest spatial resolution, but it has the enormous advantage that it enables in vivo. So we have the time resolution here. On the right of the image, we've got the highest degree of resolution. This is visualization of receptors with electron microscopy by means of immunohistochemistry. And in the middle is the method that I'll be talking about mostly today, which is in vitro receptor autoradiography. And we use it because it has the advantage of being able to visualize receptors at the microcircuit level. We are visualizing receptors in their native configuration, so exactly the configuration that they were at the time of death. And these are the receptors that at the time point of death were able to modulate the activity of the neuron that was expressing them. And it also enables, and I write very optimistically, whole brain coverage. It's not whole brain coverage in the sense of PET images, but you can do serial sections through entire brains, even through entire human brains. It's all just a question of, well, basically time and money. Now, the method in a nutshell, because we're looking at the receptors in their native configuration, we can't use chemically fixed tissue, so it has to be a shock frozen tissue. It is sectioned in a cryotome, and here you can see the example of how the sections are acquired. The sectioning procedure in itself is time consuming. I mean, it's not as bad as what Susan does, but these sections are only 20 micrometers thick, and it's unfixed tissue, so it is very, very delicate. And if you're doing these sections through an entire human hemisphere, and you reach the section around the central sulcus, this is only the occipital lobe, then you also have a very high surface to cover. Then you have to thaw mount the sections onto glass slides. And our sectioning protocol is such that what we do is we have serial sections, and from each rostrocaudal level, we acquire sections in an order which will enable us to visualize different modalities. And here I'm just showing as an example a cell body stain section, myelin section, which we also perform because we want to be able to compare our results with classical mapping procedures. And then we've got sections dedicated for the visualization of different receptors. The binding procedure itself is well, very unspectacular because the radio-labeled ligands, it's just a clear solution. But we have a pre-incubation step which is meant to rehydrate the sections and remove any endogenous substance that we have. Then we have the main incubation, which consists of two parallel incubations. On the left, the one that is labeled total binding, we only have the radio-labeled ligand, which specifically binds to the one receptor that we are dedicating this section to. And this is the total binding. And in a parallel experiment, we have the radio-labeled ligand, and we have a non-radioactively labeled displacer, okay? And this is the non-specific binding. And then we have a rinsing step which just stops the binding and removes all salts. Now we then expose these radio-labeled ligands together with standards of known concentrations of radioactivity against a film which is sensitive to radioactivity, and then we get the so-called autoradiographs. Here, these little squares, these are images of the scales with the known concentrations of radioactivity. And here we've got, just as an example, coronal sections through macaque monkey brain, where we visualize the kainate receptors for glutamate. And up here we have the sections resulting from the total binding. And down here at the bottom, there are sort of like ghost images. These ghost images are the result of the non-specific binding. And with the protocols that we use, non-specific binding is 5% or less of the total binding. So that's why we consider that our total binding is equal to the specific binding, okay? These films are like the old-fashioned X-ray films that some of you might know. So these images have to be digitized, and then they're processed densitometrically, okay? So we can just measure the gray values in these 8-bit images that we've acquired, and we linearize our autoradiographs because we've got gray values here on the y-axis, and we've got concentrations of radioactivity first in the originally digitized autoradiographs on the x-axis, but once we have linearized these images with the gray values of the microscales, which have been calibrated for brain protein content, then the x-axis really visualizes receptor densities in femtomole per milligram protein, okay? And just to make visualization of differences in receptor densities more easy to see, we do a pseudo color coding, okay? And this is how we get the images. And this slide, yes? No, that normalization is done, yes, across the section, yep. So this slide here shows a couple of examples of different receptors through an entire human hemisphere, which has been sectioned around the area of the central sulcus. The central sulcus is this one here, okay? And already at this very low resolution, you can see that receptors have a heterogeneous distribution throughout the brain. We've got regional differences, we've got laminar differences, and we've got differences between the cortex and subcortical structures. And all the slides that I'll be showing from now on have the same color coding as this slide here. So blue colors code for low receptor densities, and red tones code for high receptor densities, okay? And then to make clear a couple of principles, I'm going to zoom in to one of these sections around the area of the lateral fissure. And within the lateral fissure, we have the Heschel gyrus with the primary auditory cortex, A1 labeled here. And A1, when you look at the muscarinic M2 receptors, A1 is characterized by a significantly higher density than the surrounding areas. And the place where the cytoarchitectonic borders of A1 are highlighted by the arrows on this slide, okay? And then if we look at a neighboring section, but where we have looked at the alpha-1 receptors, you can see that alpha-1 receptors are significantly lower in A1 than in surrounding areas, but the places at which the densities change are comparable in these two sections, okay? So when two or more receptors show a border, it happens at a comparable macroanatomic or mesoanatomic even position, okay? But not all receptors show all possible borders. And on this slide here, I'm showing an example of NMDA, which at this position in the brain doesn't show this border, right? So this is one useful thing that receptors do for us. The next one is that receptors also distinguish between functional systems. And the cutout in this image is centered around the central sulcus, and on the rostral wall of the central sulcus, we have the primary motor cortex, M1, and on the caudal portion of the central sulcus, we have the primary somatosensory cortex, S1, and when you compare these three receptors, you can see that the densities of the different receptors, they're almost always opposite in these two functional systems. So when one receptor type shows a high density in one of these areas, it shows a very low density in the other area. And then if we stay within a functional system, and now I'm showing you here on the left, again, the M2, so the entire slide shows the muscarinic M2 receptors. On the left, we have a larger view of one of the sections that I showed earlier on, and you've got A1 and S1, so two sensory areas, or areas involved in sensory processing. And the image on the right is also coronal, also the human brain, but it's from the occipital lobe. And here you've got the primary visual cortex. And again, the arrows show the position at which cytoarchitectonically defined borders finish. And if we look here in the slide on the right, the visual system, you see that the density of the M2 receptors diminishes, or it decreases systematically when you move from the primary visual cortex up through always increasing hierarchical unimodal visual areas, okay? And if all of this weren't enough, because the method is fully quantifiable, we can use it also for comparative studies. So looking at different species, and it can provide us a way of really quantifying degrees of similarity of putative or homologous areas or putative homologous areas across different species, okay? And again, highlighting the fact that it is a quantitative method, and looking for an intuitive way of simultaneously visualizing the densities of as many different receptor types in a single area as possible, we came up with what we called the receptor fingerprint of the area, because we find that this balance between the different receptors in a single area, it is really as unique as our fingerprint is, okay? You can see that areas have different fingerprints, and that fingerprints can differ in shape and or size. So if we have differences in size, it means that the balance between the different receptor types is the same in the two areas, but just the area with the bigger fingerprint has a higher density of receptors, and differences in shape tell us that the areas have a different balance in the receptors, which means, of course, if the same amount of neurotransmitter is released into two different areas which differ in their balance of receptors, they're going to react in a different way. Okay, so that is why these principles are so important. Yes, Susan.
I just wanted to ask about the fingerprint issue, because fingerprints differentiate individuals of a species, but I don't think you mean that. I think you mean that they differentiate the region, or
within a region, within a species, they distinguish between different areas. Yes. Depending on the species that you look at, the fingerprint of the primary visual cortex, for example, of the human and the macaque monkey brain can be quite similar, but they are different from the fingerprint of the rat brain, for example.
So
yes.
they differentiate species,
They
not
also
necessarily
they can
individuals within the species.
Not individuals within the species. We do have inter-individual variability in the absolute expression levels of receptors, but the general shape of the fingerprint of V1 is the same across individuals.
Thank you.
Okay. So now for the next part of my talk, I'm going to be concentrating on the macaque parietal lobe, in particular the somatosensory system. So that's why here I've got S1 and S2, so primary and secondary somatosensory areas. And it's basically the work of one of my postdocs, of Meichi, okay? And the somatosensory system is basically involved in the processing of stimuli from either the outside world or from inside our body. And we know that, you know, same as any other sense, they reach the cortex via, I'm going to call them relay stations in the brain. And the question is, where do they come in the cerebral cortex? And in the somatosensory system, although we know that there is a certain hierarchy, it is not 100% understood, okay, how this hierarchy is and how the different areas are linked to each other within these different processing levels. So this is one point where we don't understand everything. The other thing is, even when we're talking about the areas involved in this processing, we still haven't agreed on the exact number and location of areas that you can identify. And I'm not going to go into detail, but I'm just showing this slide so that you understand why it was necessary for Meichi to start our project by actually mapping, creating a map of somatosensory areas, okay? And we tried to overcome, you know, this problem about the different maps by using a multimodal approach and a quantitative approach. It was multimodal because we looked at site architecture. We wanted to link our result with old maps, and it's multimodal because we also looked at very many different receptors, so 14 different receptors. And it is quantitative because we used a method developed in the lab which enables quantification of site architectonic differences and also differences in receptor architecture, and a statistical validation of the position of the borders at which these things were, or the borders were located. And if anybody is interested in the details of the method, I'm more than happy to explain it later on. And with this method, Michi identified 47 different areas within the parietal lobe, and here, just so that you can see the granularity of the areas, well, I'm showing a selection of sections through the macaque monkey brain, its coronal sections from the most rostral part that Meiji analyzed on the left side of the slide, right to the most caudal part, and the different colors code for the different areas. Okay. These areas, just the same as Susan said before, she wasn't going to, show us very many different slides, although she could spend hours looking at those images, and I could spend hours looking at your images too. I think they're beautiful, and I could spend hours looking at these images here, but we don't have the time, and not everybody can see everything, and not everybody is as fascinated by these things as I am. Just so that you see that we could validate also these statistical differences, or we could statistically validate the differences in receptor densities between our areas. And among other things, we found, for example, when moving from most rostral to most caudal sections, a significant increase in kainate densities. Yes, there's a question.
Hi. Yeah, they're great. I'm looking at these areas and I wonder how you define them. Some of them look like they, that direct, straight line might not be the best subdivisions. There's some of the receptor maybe goes a little bit more diagonal. Is that something?
So we have created straight lines because they follow the cytoarchitectonic borders. Don't worry. Yeah. No. Okay, so we don't know. We drew straight lines because these are the cytoarchitectonic borders. Receptors are visualizing with our method, we're visualizing receptors, which are mainly located in the dendrites, and we know that dendrites don't follow a straight, or not necessarily follow a straight line, so they can branch out. So this is why it could be a curved line, but we do have to,
So yeah, that's great, right? So, but in a certain way, we divide the brain in many ways, and you're now dividing the brain because of the cytoarchitectonic, which is one way, but you could be creating maps, an area based on your data directly. That's kind of what I was thinking. It's like, these are a bit separate maps. There's many ways to divide the brain in maps, right?
There are many ways of dividing the brain in maps, and as I mentioned earlier on, not all receptors show all borders. We've got receptors that merge different cytoarchitectonic areas into what we call neurochemically related areas. With these lines here, I am trying to use a way of showing these borders that other people can also use to find their areas. If I drew the borders, this is complicated enough. When you're looking, and I'm only showing 3 examples. When you're showing 15 examples and you start drawing your lines a bit to the left, a bit to the right, a curve, it starts to look like Brigitte Bardot, and then you lose the overview of the take-home message, which is we do have an area here which can be defined based on multiple modalities. Okay, so Meiji also created 3-dimensional representations of her areas. to make, because functional imaging is in 3D, because functional imaging in macaque monkeys is a rapidly advancing field. And here you see on the top left of the slide the location and extent of the different areas that she identified, represented on the Yerkes 19 template brain, and all the other small images show the densities of the different receptors that she analyzed on each one of these areas. So people can also use this data, which is freely available either by the BALSA platform or the eBrains platform. They can use it to inform their analysis if they want. And then she extracted receptor densities from all of these areas, created fingerprints for each one of these areas, and the receptors that are shown on here are such that the order of the receptors for all of the fingerprints is given by this image on the bottom right of the slide. And we could spend hours looking at these fingerprints and saying, oh, well, this one is more similar to this one when we look at this receptor or to that one. But in science, we don't want to spend, we want subjective, objective analysis, sorry. So that's why we do statistics, okay? And Mechi performed a hierarchical cluster analysis which groups areas based on the degree of similarity of their receptor fingerprints, okay? And this clustering analysis showed a clear segregation of the primary sensory areas at the top of the dendrogram from the secondary sensory areas, and within the primary areas we also have a clear separation of what is called the S1 proper, so area 3B, okay, where Mechi could also identify differences which are probably related to somatotopic differences. Now, we know that there is this link between, so this is a first inkling of how receptors can link the structure and the function, right? But the other thing that we also know is that we have different methods that can be used to link structure and function, and receptors show the hierarchy of the areas, and they segregate the functional systems. And then we have this other method, which is a truly in vivo method, okay, which is, for example, resting state functional imaging images. And what Mechi decided to do was compare her receptor data with resting state fMRI. And to do this, she used her somatosensory areas as a seed region, and then all of the maps that we looked at as, or that we have identified up to now in our group as target areas. And here I want to show a photograph of Luthia, which is another postdoc in my group, and she played an impressive, well, she basically mapped the entire frontal lobe. She's also been involved in the mapping of the occipital lobe and the cingulate cortex. So I'd like to acknowledge her work at this point here. Right, so Mechi acquired all of the receptor density data, and then she also used resting state data that is made available via the PRIME DRE consortium. And specifically, she used for those of you that are familiar with it, the resting state data that was acquired by the Oxford group, okay? And then she created a receptor correlation matrix and a functional connectivity matrix, okay? And you will see throughout the slides now, I will be using the abbreviations RC for the receptor connectivity matrix and FC for the functional connectivity matrix. Now let's look at the receptor covariance matrix in a bit of detail. What we can see is this is very clearly visible already, just like via visual inspection, that anatomically adjacent areas share numerous similarities in their receptor architecture. We also have we can also see that receptor covariance patterns are similar for areas that are at the comparable hierarchical level or belong to the same subsystem of processing. Okay, so for example, all the primary S1 areas showed consistent correlations with the secondary somatosensory cortex, with the anterior intraparietal sulcal areas and with higher visual areas, as well as with the anterior part of lateral prefrontal area 46. Whereas areas, for example, of the secondary somatosensory cortex showed consistent correlations with the superior parietal lobe and with the posterior portion of the inferior parietal lobe, okay, as well as with most visual areas and with prefrontal area 11. What Meiji also found was that areas belonging to the same system can be separated by their receptor covariance patterns, okay? And this is also important. I mean, we find significant differences in receptors. This must mean something, right? So if I show you as an example the lateral portion of area 3B, okay, we found that compared to other areas or other parts of area 3B, the internal and external portions of lateral 3B showed a greater correlation with most of the visual areas, but lower correlation with areas located in the anterior portion of the inferior parietal lobe and the intraparietal sulcus. Now, when I look at comparison of the receptor covariance patterns and functional connectivity patterns, we have both similarities and differences. And for all the areas that we analyzed, or that Meiji analyzed here, what we find is that the functional connectivity and receptor covariance is always strongest with the surrounding areas than with more distant areas. And concerning the differences, well, we can see that the receptor covariance patterns generally show or have a higher strength than the functional connectivity patterns when we're looking at the same area, okay? That for the receptor covariance, we find more negative correlations than negative connections in functional connectivity, which might be indicating differences between the areas that are really directly connected, so the structural connectivity, and the polysynaptic connectivity, which is revealed by the functional connectivity methods, okay? And with the receptor covariance, we also have we can also reveal a strong connection with visual areas in general terms, much more so than with functional connectivity, whereas functional connectivity reveals a stronger connection with motor areas. This is something that we haven't quite found an explanation for yet, but we are working we're working on it. And finally, we find that the connectivity patterns that we can identify with receptor covariance are more heterogeneous than when we look at the functional connectivity patterns. So now I'd like to move from the parietal lobe to a global view of the brain, and this is something that was done in collaboration with Sean Frudist-Walsh, okay? And what we did here was integrate our receptor data with neuronal density data, which was acquired from John Cass's group, and with dendritic anatomy acquired by Elston and collaborators. And I'm only citing one of his works here because the rest of them would fill the entire slide. And then we've got track tracing data from Henry Kennedy's data. And what we could see is that when you do a clustering analysis of the receptor fingerprints of all our areas, we can see a segregation along the first principal component analysis from sensory areas to the left to association areas on the right. And along this axis, we have an increase in the number of receptors per neuron. And when we look at the second principal component, we have a change from isocortical areas to well, I'm not showing any allocortical areas for this example, but we have shown it in the human brain, and it's the same principle, so areas that are evolutionarily older. And the differences here seem to come basically from changes in serotonin. So we have a flip here in the balance between the 5-HT1A and the 5-HT2 receptors. Now again, trying to link structure and function, the color coding of the names of the different areas here follows different functional systems that are often created by or referred to by cognitive neuroscientists. And two of these networks belong to the sensory domain, and the other ones belong to the cognitive domain. And what we see nicely is that the sensory domain is to the left of the image. Wonderful. I mean, that's what I said, you know, just in my previous slide. And all the areas on the right are from the cognitive domain. And within the cognitive domain, on the top right, we have coded in red areas of the default mode network, and at the bottom of this square coded in green, we have areas of the dorsal attention network. So basically, we are separating sensory from cognitive areas. And we are also separating areas with an internal focus and an external focus. And if you project the results of this PCA on the macaque monkey brain, the coloring, I find, it resembles very closely the functional domains that Mesolam postulated. So I think this is reassuring for us, one, to use the macaque monkey as a model, and two, because it confirms the hypothesis that this balance in receptor densities subserves functional networks. And with this, I would like to say, Go seikyo arigato gozaimashita. Thank you very much for your attention.